Hello, what's up guys? Today we are going to talk about a very interesting topic, which is genome evolution. How this genome has been evolved from billions of years ago from a single cell to a complex organism like us. So before going to the topic, so what we actually need to know is what is genome itself is. What is genome? So genome is the sum total of all the genetic material in an organism, either it be DNA or RNA. So it can be the nuclear DNA or nuclear RNA or the organelles RNA like organelles DNA like mitochondrial DNA. So genome organization contains information which encodes DNA as well as RNA. So it includes both the gene and non-coding sequences. It includes the coding sequences which can give us a protein, and it includes the non-coding sequences such as tRNA, extra. So how this genome evolved from billions of years ago till now? So genome evolution. What is this genome evolution? So this genome evolution is a set of phenomena which includes the change of structure of the genome through evolution, change of genetic structure, change of gene structure, change of number of genes, change in the content of the DNA, change in the amount of the genome. So evolution of genome includes the gene order change. Like for example, if there is a gene like this, if there is a DNA sequences, it has A, B, C, D or ATGC, adenine, thymine, guanine, cytosine, like structure, like a order of the gene like this. So if there is a change in the order like inversion and translocation, so it will change the order of the gene if there happens an inversion or a translocation. So this is the genome rearrangement. So another one is genome content or size, change in the genome content or change in the size, like there is a gain in gene. So insertion, duplication, recombination and horizontal gene transfers. Through all this, the gene can, the content of the genome can be changed, or the size of the genome can be increased or decreased. So the another one is gene loss, like deletion. It always decrease the amount of gene in an organism. So evolution of genome arrangement. So what is inversion? So inversion क्या है? Inversion is an occurrence with a chromosome break into places and the resulting piece of DNA is reversed and reinserted into the chromosome. So in inversion, like this is the DNA and this is the gene D, E and F. For example, if in a normal size like this, it is the sequence or gene order is like this D, E, F, then if there is an inversion happens, it will rotate into 183 degree. So the gene order becomes F, E and D like this. So it will give another protein, another functional protein or non-functional protein. If the structure is like this, it will give again another protein like this. So inversion evolves under selection and random drift. So the question is, can it generate any problem during meiosis? Yes, it will generate a structural problem with meiosis. So if there is a breakpoint, it can disrupt an open reading frame, like there is an open reading frame, and alter their gene expression. For example, if there is a initiation sequence and there is an ending sequence, so it will break the gene order also. So deletion consequences in some human genetic diseases, like if there is deletion caused in an organism, it will cause some diseases like autism is an example. So the types of inversion. So types of inversion that involves on the basis of the involvement of their centromeres. So this is the gene, this is the chromosome, and this is the centromere. Like chromosome structure is always like this, and this is the centromere. So if it include the centromere, yeah, if it involves, if the inversion involves the centromere, we call it pericentric. And if there is the non involvement of this centromere, then we call this type of inversion as paracentric inversion. So what's translocation? Coming to the topic, translocation. So translocation is the transfer of a piece of gene from one chromosome to one homologous chromosome to the another chromosome. So it is actually the rearrangement, the part of chromosome between non-homologous chromosome from one chromosome to another chromosome. So if there is a gene translocation process happens, it can be constant process throughout bacterial genome, that and it suggested that the translocated gene are deleted rapidly during evolution. So this is an example of translocation. What we are seeing here is, this is another chromosome, chromosome 4 and chromosome 20. This is before translocation. See, after a translocation, the piece of chromosome at the end of chromosome 4 is break down and transferred to the chromosome number 20. So the another piece is, this is the piece of chromosome 20, and the half, the small arm of the chromosome is the chromosome 4, the piece of chromosome 4. So we call this chromosome is Philadelphia chromosome. So this is a translocation. So how the gene evolve in a genome content. So how the evolution of genome content varies or it change. So there are generally two types, like one is gene gain, the another one is gene loss. So if this process like insertion, duplication, recombination, horizontal gene transfer, sequence diversion, if this type of process happens, then the organism will gain some gene. So these are the source of gene acquisition. And the another one is gene loss. If there is a deletion happen in an organism, then the gene will loss. For example, if an organism genome has 25 genes, if it deletes 2 genes, so the genome of the organism become 23. So the another topic is, so I'm explaining all these things. So gene gain, how the gene gain through the process of insertion. So insertion, what is insertion? Insertion is the addition of one or more nucleotide or base pair into a DNA sequences. So if there is a DNA sequences like this is the original DNA, CATCA like this, the long chain. So in nature there are some mobile elements, or we call transposons, or some other insertion sequence exist. These encode the information which is necessary for the insertion of the DNA or nucleotide base pairs. So insertion, so there is a TC and A, see here TC and A, but there is an insertion of one single nucleotide adenine. So if there is a normal protein happens between TC and A, this code for an amino acid, for example, if it gives a protein tryptophan, for example, so if there is insertion, the sequence become TAC, so it will give another protein like tryptophan. So the chain of amino acid sequence will change. It will give another protein, or it will give a stop codon or a start codon, like in an open reading frame, if there is a start codon and if there is a stop codon. So if there is another protein, for example tyrosine, and at that place it gives a stop codon also. So what is genetic recombination? So recombination is a process by which the molecules of the nucleic acid, generally it is DNA, sometimes it could also be RNA, is broken and then joined into a different one. So recombination can be dominant force in shaping genome in associate with the phenotypes. So it will not affect, recombination generally do not affect the genome, it will change generally the phenotypic characters, जो हम आंखों से देख सकते हैं वो characters में change हो जाएंगे. So horizontal gene transfer is also called lateral gene transfer, क्योंकि जब इसमें gene transfer होते हैं, the gene transfer are between organism other than vertical gene transfer. So vertical gene transfer क्या होते हैं? It occurs in the exchange from parent to generation. For example, horizontal gene transfer. This is horizontal gene transfer from one organism to another organism. So vertical gene transfer is parents to offspring, from parents to F1 generation. So the gene from parents which transfer into the inherit into the F1 generation or the offspring is called is called the vertical gene transfer. So from one organism to if another organism it happens, then we call is horizontal gene transfer. So what is gene duplication? The gene duplication or chromosomal duplication or is generally called gene amplification. So when a gene duplicates, it can make a number of copies of the same gene or same DNA. So the gene duplication can happens or can contain at any region of the DNA that contain a gene. So gene duplication is believed to be play an important and major role in the evolution process because it is an example of yeast genome. So yeast genome has been evolved with yeast genome has been underwent a duplication about millions of years ago. So generally the plant plant genome are a prolific duplicator. They duplicate at a very fast and rapid time. For example, we can take a wheat is hexaploid. So see if the duplication from haploid, polyploid and all the 6 times. So the gene duplication could be orthologous or paralogous gene. So what is orthologous gene? Orthologous genes are two genes which has been diverged due to speciation events. So in two geographical areas, if there is a two organism which remain in due to certain climates, they divide and diverge in into two geographical areas. So the another one is paralogous gene. So these are the two genes. We believe it to be say that the paralogous genes are the two genes if they diverged after a duplication event. So the orthologous gene is if there is a population, so the same gene has been diverged due to speciation, another one remains here, the half remains here. This is duplication. And if there is a gene, it make a number of copies, copy 1, copy 2, copy 3. So these all are diverged into separate times or separate locations. So we call this a paralogous gene. At first we have the same function, because these paralogous genes are duplicated genes. They could have the same function, but due to environmental change, due to remain in the different environments, their function may varies with time to time. So this is an example of this is a flow diagram of gene. What is homologous gene? Homologous is the same gene. Like if two genes are diverged from one due to speciation, we called as orthologous gene. Orthologous gene. And the same genes due to duplication, if their genes become another genes separated, so we call them paralogous gene. The example of paralogous gene is the globin genes, early globin genes. They have only alpha chain, so the alpha chain, and they divided into two, duplicated into two gene, alpha and beta gene. So basically their function remains same with time to time, but with time to time due to some mutation or another environmental factors, their function can vary. So generally there are two ways in which gene could acquire by the genome: by duplicating some of its existing gene into the genome, so it will increase the content of the genome, or by acquiring gene from another species like so gene duplication contributes to genome evolution. So how these gene duplication contribute to genome evolution? So there is a hypothesis called it gene duplication hypothesis. It believed that the mutation can cause duplication in gene, which allows one copy of the gene to mutate and evolve to perform a new function. So if there is a mutation, it happens to be duplicate a gene, so the gene the copy, so while allowing the other copy of the gene to continue to perform the original gene function and the original gene, and the duplicated copy is believed to be the main source of all new genes. So what is pseudogenes? Pseudo means false. Pseudo is generally we can call say false gene. So it's not false gene, it's actually a gene. So the pseudogenes are the gene in a genomic DNA sequence which is very similar to the normal genes, so but they are non-functional, so their functions are actually silenced. They are related, we generally call them different relatives of functional genes. So duplicated genes may diverge to generate different genes, or one copy may become active or inactive pseudogenes. So pseudogenes are very important since they provide a record to how the genome has been evolved, how the genomic DNA sequences has been changed from time to time. So it is generally used for a model for determining the rate of nucleotide substitution, insertion or deletion of the sequences in an entire greater genome. So some example of the pseudogenes are actin ferritin genes, and another gene is glycerol dehyde 3-phosphate dehydrogenase. These genes are some examples of pseudogenes. So what are the types of pseudogenes? So on the basis of their splicing, we can divide the pseudogenes into two genes, two components like processed pseudogenes and non-processed pseudogenes. So generally in the gene processed gene and non-processed genes are when there is a sequence or DNA or mRNA sequence, if we remove the exons, not exons, remove the introns, then we call it as processed genes, or the process of splicing happens, then we call it as processed genes. So the processed pseudogenes are the results from where the reverse transcription and integration of mRNA transcript. So the mRNA, after the mRNA formation due to reverse transcription, it become DNA again, then pseudogenes, they are the duplicated genes and they become a pseudogenes. So these are the processed pseudogenes. So non-processed pseudogenes are those genes which have intron in their genome sequence or intron in their DNA sequences, which are resulted from incomplete duplication or secondary copy mutation of the same functional gene. So the pseudogenes, some pseudogenes may gain function differ from those of their parent genes, such as the regulation of gene expression, and take on different names. So gene duplication contribute to genome evolution. Generally we say that how this happen, because duplication occurs at 1% of the gene per million year. So the divergence accumulate almost 0.1% million year. So active, or it become silencing of one copy takes 4 million year almost, so it becomes active or it become a pseudogene. If it is inactive, then it is a pseudogene. So we can take an example of the globin genes. So alpha, beta and gamma, alpha, beta, delta, all these are duplicated from a single genome and accumulated between their copies, and they make a number of copies. So this is the classical model of fate of gene duplication. This is the normal gene, so normal DNA sequence or gene, and it duplicates and make a 2 copies, so 2 copies of the same gene. But if these become a non-functional gene, then we call them pseudogene, or it gives a new functional gene. So it gives a new protein, or it gives a new sequences, so it is generally called it neo functionalization of the gene. So how organism acquire new genes from other species? So a genome or an organism can acquire new genes from another species. So as comparison of bacteria and archaeal genome sequences, it suggested that lateral gene transfer has been a major event in the evolution of prokaryotic genome. So there are two, I already said that in the first slide, that there are two types of gene transfer, like one is lateral gene transfer and another is horizontal gene transfer. So the horizontal gene transfer is from parents to offspring. So the lateral gene transfer is from one organism to another organism. So this happens with the few processes such as transduction, transformation and conjugation. So what is transduction? Transduction is a process by which the DNA is transferred from one bacterium to another bacterium through a virus or through a bacteriophage. So in conjugation में होता क्या है कि जब कोई बैक्टीरियोफाज एक बैक्टीरिया को इन्फेक्ट करते हैं तो वो अपना प्रोटीन बना लेते हैं, तो लास्ट में कैप्सिड में वो जो बैक्टीरिया का डीएनए उसमें क्या करते हैं, फिक्स करते हैं, गलती से हो जाते हैं, तो वो फिर दूसरे जब बैक्टीरिया को वो इन्फेक्ट करते हैं तो उसका जो जीनोम है या उसका जो डीएनए है उसमें ट्रांसफ़र हो जाता है. सो कंजुगेशन ऑल्सो इनेबल्स to move plasmid between bacteria frequently to acquisition of new organisms. So conjugation में generally एक क्या होते हैं, उनके अंदर एक लिंक बन जाते हैं, एक लिंक बनाते हैं, सेक्स पाइलस कहते हैं जिससे हम उसके थ्रू बनाते हैं. सो अनदर प्रोसेस ऑफ डीएनए ट्रांसफर बिटवीन प्रोकैरियोट्स transformation is more likely to have an influence on the genome evolution. So this is the process how gene transfer from one organism to another organism through lateral gene transfer. So the first process transaction, as I already said, that it happens due to bacteriophage. See, these bacteriophage have infected one bacteria and at the end in their protein capsid they collected not only their genetic material but also the some pieces of DNA from the bacteria which infect another bacteria, and they transfer the another cell. Like this is the donor one and this is the recipient one. So in conjugation what happens is there is a formation of an conjugate, there is a formation of a pipeline structure like sex pilus from one to another bacteria, to donor and recipient. So in transformation is the transfer of free DNA. So one bacteria can due to damage or incompetence the genes are transferred into the air or into the environment. So another bacteria which is donor bacteria can accept or receive that DNA from the environment. So coming to the molecular clock. So what is molecular clock? Molecular clock is a technique in the molecular evolution which always use fossil as a constant and the rate of change in their molecular or DNA structure which deduce due to the geological time history. So it is used to estimate the time of occurrence of event called its specification and radiation. So molecular clock generally use the molecular data to calculate the nucleotide sequences, change in nucleotide sequences in the DNA and change in the amino acid sequences in the RNA, not RNA, in the proteins. So a constant rate of sequence divergence is in molecular clock. So the sequence of orthologous gene in different species varies at non-synonymous site. So what is synonymous and non-synonymous site is if there is a gene where mutation have caused amino acid substitution. So if there is a hydrophilic amino acid, the same hydrophilic amino acid has been happened after the mutation, that's called synonymous. If there is a hydrophilic amino acid and due after mutation or substitution there is another protein came or another amino acid which is hydrophilic or hydrophobic, so it affect the amino acid sequence actually. So synonymous substitution accumulate 10x faster as compared to the synonymous divergent or substitution. So molecular clock hypothesis, what this hypothesis says is the evolutionary divergence between two DNA sequence can be measured correctly, corrected percent of position at the corresponding nucleotide differs. So the substitution may be accumulated at more or less constant rate after gene separate. So therefore the amount of genetic differences between these sequence is the functional time since the separation. So rate of molecular change is not inconstant, it's constant enough to predict. So we can say this amount of DNA or this amount of nucleotide has been changed or has been diverged. So to measure the evolutionary time with a molecular clock, we need to use certain formula like this: T is equal to D by 2R. So that D is to estimate the genetic distance, number of amino acid replacement, how many amino acid has been replaced. Like in a chain of DNA, if there is a change in sequence, the change in amino acid sequence in the mRNA will also change. So they use the paleontological data to determine the data of common ancestors. So we take the T is equal to time since divergent, and to estimate the calibrate rate, so number of changes expected per unit is R is equal to D by 2T. So the divergence in alpha globin genes, for example, the millions, 100 millions of years ago, there is only a globin genes. So due to the fusion, duplication and divergence, single globin genes, there is only a single globin gene in lamprey and hagfish. Interstellar globin was myoglobin. So after that, the duplication and divergence, it become alpha and beta gene, which is linked to the amphibian and fishes. So another, they separate with time, the evolutionary process, the series of duplication and separation happens, that alpha becomes separate and beta becomes another. So primarily their function remains same, to carry the oxygen in the blood. So after few billions year, that the gene has been expression of clusters, so it again becomes alpha 1, alpha 2, beta 1, beta 2 in humans and animals, not humans and mammals and birds only. So the another one is mobile elements. So diverse of gene evolution, there are mobile genetic elements. So these elements are, these sequence are you can move from run within the genome from one place to another place. So it was first discovered by Barbara McClellan in Mays in 1950. So she has challenged the idea that genome were a static entity. So major diverse of genome evolution has happened. So this mobile genetic element is we can call as transposable element also. So mobile cassette also. So what is this transposable element? These are the sequences of DNA which can change its relative position, self-transposon, with the genome of a same cell, single cell, not from one cell to another cell. So the mechanism of transposition can either be copy and paste or cut and paste. So phenotypically there is a significant mutation, and that can alter or change the cellular genome size. So the types of transposable elements, one is class 1 type and the other one is class 2 type. We said that there is a copy and paste. So copy and paste is they make their copy from DNA. There is a gene, for example. So here is ATGC. It is a transposable element. For example, it become mRNA to again DNA, so it become copy and paste, and they insert it into another position like here. So this is the class 1 transposon. So first transcribe the DNA into the RNA or mRNA, and then again with the use of reverse transcriptase enzyme it become DNA again and inserted into the another position in the same cell. So it's called reverse transcriptase. The enzyme is called reverse transcriptase, and the process is called reverse transcription, which make a DNA copy of the RNA to insert into the new location. So the class 2 transposons consist of cut and paste. It generally cut the same DNA over here using some enzymes and inserted the same DNA over here without transported into the another organism. So retrotransposons in genome evolution. So I said that copy and paste mechanism is the retrotransposon. We call them retrotransposons. So they make RNA before DNA, then again using the reverse transcriptase enzyme they convert RNA into DNA and insert it into the new location. So the enzyme is called reverse transcriptase. So many retrotransposons have long terminal repeat LTR at their end, long terminal repeats at their end. So larger genome may have many retrotransposons which are actively contributed to genomes. Some example of retrotransposons are copia element in Drosophila melanogaster and TY elements in yeast. So this is the process of transposition, like retrotransposition. So this is the normal gene, so it can transcribe into RNA, mRNA. This is called transcription, and after, if it happens to be translated, then some protein, it makes some protein or enzyme called reverse transcriptor due to with the process of translation. So this protein, this enzyme are using this enzyme reverse transcriptase, the same RNA will convert it into the DNA again and synthesize a second strand copy, and this DNA will insert it into the another position or insertion. We can say insertion of retrotransposon or retrotransposition DNA. So how transposable element contribute to genome evolution? So the multiple copies of similar transposable element may facilitate recombination or crossing over between different chromosomes. So insertion of transposable element within a protein coding sequence may block protein production. For example, if there is an open reading frame, so it has a initiation sequence and at the end it has a closing sequence or stop codon. We can say stop codon. So initiating codon till stop codon, between them if there is a transposition happens, if transposition happen on that position, so the DNA, the sequence, if there is a stop codon coming in between, then the production of entire protein will not be happen. It will stop and will break in between of them. So insertion of transposable element will affect the protein production, and it also affect the rate of protein production. It can increase the rate of protein production, and it can decrease the rate of protein production when these transposable elements inserted into the regulatory side, like into the regulatory site which regulate the protein synthesis, like so transposable element may carry a gene or a group of gene in a new location. So you can understand this. So it can create a new site for alternative splicing in RNA transcript. In all the cases, the determine and occasion proved advantageous to. So example of transposable elements are ACDS element in maize. This is an example of transposable element. So role and effect of transposition on genome. So transposable elements, it tends to increase the copy number with introducing introduce it to a genome. So when a transposable element enter into a genome, it can increase their number of copies. So transposons are generally mutagens. It can cause mutation. So transposon are retrotransposons. When it inserted into a gene, it is insert into a functional gene, which can disable that gene also. So in exon and intron, there is a flanking region at the end of both. For example, which enhance the which may contain the promoters and enhancer. So these are the regulatory site. If they inserted in this region, they will change, they will regulate the increase and decreasing amount of protein in that gene. So hemophilia is, or A and B, severely SCID, are some Duchenne muscular dystrophy. These are some diseases which are related to the transposable and which are happen due to the transposable element. So this is an art of mobile genetic element caused this prized color pattern in morning glory. See, this is the color difference here, and it will entirely change at that protein phenotypic change. It happens at the level. So C value paradox. What is C value paradox and why eukaryotic genome size varies? So C value paradox is the amount of DNA. So not C value paradox. What is C value? C value actually is the amount of DNA in an genome. So haploid per set of the chromosome. So C value is the total amount of DNA in the genome. So the entire DNA in a genome is called C value. But the lack of relationship between the complexity and amount of DNA in higher eukaryotes, which the phenomena is called C value paradox. So generally in haploid organisms like mycoplasma, bacteria, reptiles, in these organisms, in lower eukaryotes organism, when their genome complexity increases, when the DNA size increases, the amount of DNA increases, the complexity also increases. But in higher eukaryotes, there is no link between, there is no relation between the amount of DNA and complexity of genome. So that phenomena is called C value paradox. So what is adaptive evolution? Adaptive evolution is an evolutionary process which is based on creature need or need within an environment. If there is two organisms happen to be on a same site, bound to be on a same site, so if they go into the different environments, and according to that environment their structure will start developing. For example, camouflage techniques in snow region bird and forest bird. For example, if the bird happens to stay or live in a snow region, their camouflaging techniques will develop according to that environment. And if it stay in forest, their camouflaging techniques will be based on that region also. So the association of an increased rate of gene gain or positive selection is generally an indicative of adaptive genome evolution. So here is an example of adaptive evolution, like single nucleotide polymorphism SNPs, which affect the beta globin gene, which results in curved and elongated RBCs and are removed from the circulation. So a homozygous mutation usually die from anemia without intensive care. So heterozygous individual will have mild anemia but will deal better than the malarial parasite Plasmodium falciparum, maybe because infected red cell become sickle. So the malarial distribution history, so in this region, so if the organism have heterozygous individual which have mild anemia, so because their the normal cell is like this RBC, so due to anemia or due to their due to the SNPs, change in SNPs, the cells become, the RBC become sickle shaped, which we also call sickle cell anemia. So mitochondrial genome evolution. So coming to the topic, mitochondrial genome evolution. So mitochondria seems to be a monophyletic in origin. So according to the symbiotic theory, so the mitochondria and chloroplast are endosymbiotic coming from a bacteria which can make a symbiotic relationship with the plants and animals, and they become the part of this. So studies of early divergent protists generally find a minimal divergence in alpha protobacteria, similar with alpha protobacteria. So it is thought to be that the alpha protobacteria are relatives of mitochondria. So what it implicates is their implications of phylogeny of eukaryotes. So evolution of plant mitochondrial genome radically different from the animal mitochondrial genome. So ancestral and derived mitochondrial genome. So in ancestral there are many genes. So the bacteria like RNA genes was present in the ancestral mitochondrial genome. So complete set of tRNAs were present. So mostly coding sequences were present. So the gene cluster was also present in there. So standard genetic code was also present in the early or ancestral mitochondrial genome. But later on, when after the endosymbiotic theory, when it became an organelles of the eukaryotic cells, the loss of genes happens, divergence of RNA, ribosomal DNA. So high mutation rate was happen, non-coding DNAs was absent. So so biased codon usage was also happens over there. So non-standard genetic, it becomes standard to non-standard standard genetic codon. So diversity of mitochondrial genome. So in animals, fungi and plants, the size is in animal 14 kb to 42 kb. So this is the diversity, this is the difference in plant, animal and fungi mitochondria genome. So in fungi it is 17 kb to 180 kb, and in plant it's 184 to 2400 kbs. So the non-coding DNA present in animal is very low, it is very absent. So in fungi it is variable, and in plants it's very high. So the mutation rate in animal mitochondrial genome is very high, and in fungi it's very low, and in plants it is very low. Exactly. And in the recombination is not present in animal genome. So in fungi it is sometimes present, it is variable. In some fungi it is present and it's not present also. So in plants the recombination also occur in mitochondrial genome. So introns are not present in their mRNA sequences in their protein sequence in mRNA sequences in animal. In fungi is not present or absent, in plant it is present also in trans. So universal genetic code is not present in animal, because in animals it completely become an organelle of the cell. So in fungi it's mostly available, and in plant it is completely available. So what are the characteristics of mitochondrial DNA? So the first characteristic of mitochondrial DNA is that it inherited from the mother only. So to an offspring, your mitochondrial DNA is derived from your mother only. Your father cannot inherit or give you the mitochondrial DNA. So the function we can use by using the mitochondrial DNA, we can trace the evolutionary linkage through the maternal links also. So the mitochondrial DNA is generally circular. Mitochondrial DNA is generally circular, and it is double stranded. So it's like this. Double standard mitochondria. So their size is actually 15 to 18 kb. So numerous mitochondria per cell can be happens, and there is almost 500 to 1000 mitochondrial genome per cell are present. So evolution of gene with novel function. So gene evolution with new function. The copies of some duplicated genes which are divergent in a much evolutionary history, their function can include it with different proteins or different function. It can become non-functional also. So the so after divergence, the encoded proteins are now very different. For example, there is the lysosomal gene. So lysosomal gene after duplication, it becomes the alpha lacked albumin genes in mammals. So lysosome is actually an enzyme which protect the animals from or against the bacterial infection. So after duplication, it gives alpha lactalbumin, which have a entirely different function, which is actually non-enzymatic protein, which plays an important role in milk production in mammals. So comparing genomes. So genome comparison of closely related species help us to understand what. So if we are comparing the genome of one organism to another organism, if we compare the genome of our closely related species, it will help us to understand the recent evolutionary events or recent evolutionary change happened between us. So the genome comparison of distantly related species will also help us to understand ancient evolutionary events. So the relationship among species can be represented by tree-shaped diagram. So we can call this evolutionary tree or evolutionary history of an organism. So comparing closely related species in their genetic differences are very between the closely related species are very low, and we can correlate the phenotypic differences in between us, because our phenotypic characters will almost same, but there is a little differences because of a little evolutionary change, because they are our closely related species. Our DNA, our sequences in the DNA are almost same, with the exception of few nucleotides. So let's take an example of genetic comparison of several mammals with non-mammalians to identify what it takes to make a mammal from an amphibians to birds to mammals. So let's take an example of human and chimpanzee genome. So it only differs between 1.2% at a single base pair only, and by 2.7% because of insertion and devolution. So in generally we says that humans have been evolved from chimpanzee, so their genome and our genome has only 1.2% differs in a single base pair. So several other genes are evolving faster in human than chimpanzees, so we become so mature, and we almost the evolutionary change or mutation happens in human faster as compared to the chimpanzee, which includes the gene involvement in defense against malaria, tuberculosis, regulation of brain cells, and genes that code for transcription factors. In these genes or in these region, the evolutionary change or the mutation has happened faster in humans as compared to the chimpanzee. So we are coming to the conclusion. So I am concluding this lecture at by saying this: the rate of genome evolution is not constant. It can differ from organism to organism or differ from time to time. If there is a high radioactive environment or high different environment, it the rate of evolutionary genome or evolutionary change will differ from one environment to another environment. So the rate of gene acquisition has increased. The rate of gene acquisition, lateral gene transfers, gene acquisition due to duplication, and all this has